Good morning, everyone. It's my pleasure to be moderating this meeting regarding the vascular development in the brain. My name is Mehdi Jorfi. I'm a neuroscientist and bioengineer at Mass General Hospital in Boston. And without further ado, I'm gonna introduce our first speaker. It's my pleasure to introduce Vanessa from Seattle Children's Hospital. She obtained her PhD degree in Portugal and then moved for her further studies in the United States. Vanessa, the stage is yours.
Thank you so much. First of all, I want to acknowledge the organizers. I think this is a great initiative for promote and have a boost in our new studies. So thanks for having this and put all this together. Okay, so I'm going to talk about how brain vasculature forms in the brain. So brain is the most energy demanding organ in our body, although they lack some supply of energy on the energy storage. So they maxed up the brain vital dependence on the vascular system. So I have the blood flow supplied by arteries and distributed by a network of capillaries and drainage by vein. In fact, this network of capillaries and microvasculature is remarkable in several ways to along this perfusion of the blood. Is organized in such a way that every cell is no more than 15 microns away from the capillary, so allowing this, and densely packed into all this brain tissue for efficient nutrient and oxygen delivery, but also a waste removal. Third, but not the least, is also the ability to form the blood-brain barrier, a barrier that protects brain against pathogen, but also allows this exchange between blood and brain. Oops. We have two waves of forming this vasculature in brain. We have the vasculogenesis, that is the de novo formation of vessels and normally occur embryonically. And you have a second wave of formation of vessels that occurs by the formation of new vessels by existing vessels, and it calls angiogenesis. And normally these occur postnatal. Also during this timeline, postnatal is when you occur the assembling of the cells that compound the neurovascular units that you have endothelial cells, pericytes, astrocytes, neurons, and microglia renew all together to form this beautiful friendship and make this united work functionally. So we have homeostasis in the brain also that allow the communications between the brain and the blood occurring normally, and you have as you have in adults. Besides all the studies that you have that allow us to understand how and what the pathways involved and necessary for the formation of these vessels, what is the protein critical, we miss a lot of the dynamics of this process because a lot of this critical work and necessary work was being done histologically. So we miss some dynamics in the mammalian brain. So I have a lot of questions that you cannot answer using this approach. So namely how the vessels arise. They come from veins and arteries in the same way. So we have this schematic that you have sprouts coming and all the spot coming and they found together to form. This is also true in the mammalian brain. Also, how the blood supply cannot be interrupted in the extensive growth when you have these new vessels forming and connecting to perfuse distant vessels. So if you think that you have like new roads forming, you can stop the traffic, but like in brain, you cannot stop in the flow. You need to keep the blood flowing in order to have a function development and function brain. Also, how this highly redundant and interconnected architecture is forming the capillary plexus. Also, we know the pericytes is needed for stabilized vasculature and needed for quiescence, how they can stimulate and simultaneously provide plasticity in order to form new vessels. Are endothelial cells leading or trying to be guided? So all these questions that needs a dynamic, they are involved in the system. So we need new methods for studying this dynamic process. So the aim of this work was really like groundbreaking this and trying to understand and characterize the postnatal cortical vasculature remodeling process, how this relates with the basal cerebral blood flow. For that, we refined existence protocols in the literature for live imaging in neonatal brains to allow this be longitudinal. So we shave the bone very carefully using a razor. So we have these cranial windows, and we observe the pups. They develop normally. We did not observe alterations in weight. The dams carry them the same way as the non-surgery pups. And also we did not observe any alteration in behavior. Taking account also the studies that were being done histologically, we focus our attention in expansion periods. So we performed the surgery P7 and we did daily imaging until P12. This corresponds roughly between 36 weeks of gestation and initial months after birth in humans. So I started like visualize this dynamic process using a type 2 GFP animal. So we know a lot of signaling is involved in angiogenesis, and type 2 then angiopoietin 1 is one of most important, or at least if you knock out this pathway, you have several abruption in the formation of vessels and can be lethal. So this animal express GFP correlated with the expression of type 2. So we can follow the expression of this type 2 over time. But you can see here the type 2 in green, and also inject IV dye with high molecular weight. So we can see also how the vessels are perfused or not. So in this case, you need to see both colors at the same time in order to understand how the new vessels are formed, because if you inject IV dye, the new vessels are not perfused yet. So here, to be simple, we have the two colors merged, so the type 2 in the IV dye and the black and white modes. And as you can see, everything is changing in more than one day. And believe in me, this is the same field of view. So we have some clues. So we have these penetrating vessels that you can see in here. Observe it. So you have some clues that you can observe. And what we did was drawing. So we have this penetrating vessels by these round balls, and what is already formed in black, and the new vessels in green, and connections, new connections in orange. So we quantify this over time, and you really observe that corroborating the studies that have been done in histological approaches, that we have an increase of the sprots and have a peak at P9, and these connections are being made and have a peak at P10 and they go down until P12. So it's really like the expansion period that we are looking for. Next, our question was, they coming from veins, arteries, both. So to distinguish veins and arteries in vivo, we have here in the surface of the brain, PL surface, with pseudo-colored venules in blue and arteries in red to be easier to you identify them. And you can identify by morphologically. So arteries are much more straight than veins are this tortuous kind of shape, but also can by line scans. So if you draw a line scan and the direction of the flow was opposite to the direction of my line scan, means that the blood is coming out from the brain. So it means it is a venule. And if you draw this line and the direction of the flow is the same of my line scan, means that it's going inside the brain, so it's artery. So we did exactly the same approach. So we have the same view and you follow over time. But here, when you draw it, we add venule or arterial side. And again, we draw the sprots. And we found that all the spots in all the time, in all the animals, they came from veins. And so we have here like quantify over time several animals and never saw any spot coming from arterial side. Our next question was, are this organization, there is some organization in the cows. So here, to be simple, I have in black what it's supposed to look like in adult's brain. And so what I mean by order is close to the Shannon Venel, or so we have order 0, a Shannon Venel, and 1, 2, and 3. So what happened is you really have this growing in an organized way. So in order to have the order 2 presence, you need to have the order 1 formed before. And I guess our next speaker probably going to speak this in more detail, but like in retina, you have these fronts and geogenic fronts and everything is like formed, but you know, you need to set up first the stage in order to have another spot coming from this order. In fact, when we went to analyze this in detail, we noticed 2 different sprots in terms of morphologically and also dynamics. We found that there was some sprots that connect and targeting arteries and others they connect intercapillarily. So here you can see like an example coming from the vein and it's growing, not perfused and connected to artery. And here like shortenly connects intercapillarily. So it's really, when you look and measure, we could like classify them as a long range arterial targeting spots because they are much longer and they grow much more than the ones that connect to capillaries that you call like short range capillary targeting spots. We even break these more to try and understand because we observe this state of formation that you have order 0, order 1, and so go on, if that can be related with these different types of sprots. And in fact, there was. You have these long range sprots, they form from the venous branch order 0 or 1. So as you can see here, and quantify in this graph, but also they arise much early than the ones that connect to and form these short connections between the capillaries. So it's really like an organizing way that you form this architecture inside the brain. As I explained in the beginning, like parasites and endothelial cells cooperate together to form this microvasculature. And there is like these debates are how the parasites can be at the same time formatting the quiescent and be essential for various properties, but also provide the plasticity to new vessels. Or perhaps you have the formation of the vessels and then you recruit the parasites. To answer this question, we have a triple transgenic using the type 2 GFP and the PGFR beta. So you can see the endothelial cells, the sprots, and at the same time you have the parasites marker as well. And you observe all the capillary beds, or at these formations, the parasites are already there. You didn't see a reservoir available of sprots without parasites. All of them express already. In fact, these sprots normally arise from places from soma with the parasites. And probably what you think about is like that is a multiplication of the parasites along the way, and they go to form the vasculature. As you can see in this sum over there, the spot is coming here, multiply the sum of the parasites, and then when it connects, the sum will go back, and the same over there. In fact, as I say, we observe the parasites are already at the sprots, although the localization seems a bit different. So we observe there is some sprots that were completely covered by the parasites, and others where the parasite is a bit behind of the sprot tip. And when you look at the sprot length, it seems like that the ones that hide behind probably are the ones, the long, arterially connected ones, and the short, the ones that connect to capillaries. And in fact, it seems like that. So with the ones that are behind are the ones that go to connect to arteries, and the short, that are totally covered by the pericytes, are the ones that go and connect to the capillaries. It makes sense with the fact if we believe they are quiescent, so they promote the less growth in the case of the capillaries. Could be some indication. Last, we're trying to understand how this remodeling could affect the cerebral blood flow. It's still interrupted, how they grow, how the blood flow relates with this growing. First, we look to the surface of the brain and all the penetrating arteries, so the entrance of the blood, and also the number of ascending veins, so the exit of the blood, did not change over time. So it also allows us to get these clues, so the places, because they don't change as much. So it seems like this remodeling and the change in the blood flow really depends from the capillary bed expansion. Because of that, we measure the diameter of this penetrating vessels in order to understand the blood flow, because the spots are not perfused, or sometimes they are not flowing, so you cannot measure the data from that. So what we did was measure overall both what is coming in and what is coming out, to have a bigger picture of the blood flow over this timeline. So we observed that there is a state, there is no change in the diameter in the ascending venules, as you can see by the yellow lines here. So it represents how the first diameter in the P8 is not changing, although in the arteries you have an increased widening of the arteries, probably because they also are remodeling, so the musculature is forming elastin, so over time. Additionally, you also measure the velocity at these vessels. And in here, opposite, we have an increase state of velocity in ascending venules, but not as much as the arterioles. Put all this together in this formulation, so diameter, velocity, over time, you observe an increase of the flux in both, more pronounced at the arterial side. Okay. We also put all these parameters together: the angiogenic spots, the capillary connections, the arterial flux, venal flux. So all this normalized to try to understand what is really driving these alterations in flux. And it seems really driving by the capillary connections over time. So you have more capillary, you still have more fluxing going in and going out, but in a very organizing way. So you never have interrupted dead ends at this growth. So as conclusion, I summarized the paper here, and the take-home message that I want to leave you is the cerebral venules are the locus of this angiogenic activity in developing brain, and probably, who knows, could be a potential target for modulation, endogenous capillary growth, and repair injury, but also maybe could be the ones that cause this arterial malformations, because now we know they are the source of the angiogenic spots. Maybe they can also be somehow modulating these arterial malformations that sometimes you observe in the brain. Also, we've observed two forms of vascular growth. They're resulting in this equiescently defined architecture branching in organization of this capillary bed. So you can answer some of our questions. And this bulk of capillary expansion occurs in a concerted way with the widening of the penetrating arterioles, but also contributes to a steady increase of the cerebral flux. So I want to thank my group, SHE Lab, we are brain imagers, all the collaborations, all the funding, and of course you for your attention. Thank you so much.
Thank you, Vanessa. That's for fascinating presentation and very interesting work. For the audience, please cast your questions in the chat room, or you can just unmute yourself and start asking your question. Okay, I can start with a few questions. I have several questions to go with this. One thing I was thinking about the astrocytes, where the astrocyte fits in the blood vessels development.
Yeah, this is a great question. I have the same one, and I hope maybe I can answer that in a couple of years. That is, they grow in the same time. So the timeline we have in some studies, they seem to occur in the same timeline. So in between the first 2 weeks, although it's not as much studies in postnatal how they enwrap and what is the timeline of these enwrapped vessels. So that is not that much known. Yeah. So I believe there will be, it's really interesting study as well.
Wonderful. Robert?
Yeah, thanks, Vanessa. Very interesting work. Say hello to Andy for me.
I think he's here around. Know his name.
Hello, Andy, if you're there somewhere. Question about depth. So I don't know that I haven't read your paper carefully, but did you see any differences? So you're obviously constrained a little bit by where you're imaging.
Yes. So because it's PAPSIN's a thin skull window, you cannot go as deep as a chronic window. But I could, in like the best windows, go until like 350 micron. So it's quite a pretty good deep. But also what happens in the growth is I need to go every time deep more or less 50, 70 microns to find the same spot. So the brain is growing in depth and also expand like a bit to the left. So it's expanding in both ways. So we need like really found these clues. So when we found that the penetrating arteries and venules didn't change, for us was really like, okay, I can found the same spots by these clues. But yeah, we have limits a bit for in depth.
And did you examine sprouting in leptomeningeal vessels, like at the cortical surface versus intraparenchymal?
At the PL service, we found that was a pruning of the venule. We also have that at the paper at the same time, although they occurs much early. So some of this pruning occurs in TP6, and then you have less, but we didn't find like sprouting above. But we don't know if like probably some of the lymphatic still forming because it's postnatal, but we didn't look at that.
So I guess the last question for me, Mehdi asked about astrocytes. I'd ask about other cell types, around the peel vessels. So it'd be very interesting to look at that too. You mentioned lymphatics. There's a lot of discussion around that.
Yeah,
the development of those systems. Yeah.
Yeah, we are collaborating now at a group in Colorado for doing some of the experiments with the fibroblasts. So, but will be like a collaboration for complete their studies, but it also gonna give you some insights how they move from the surface until underneath. So one more perivascular cell to join the club.
Yeah. Thanks very much.
Thank you.
Wonderful. Any other questions? I think someone raised. Okay, Gina, yes.
Thanks. Yeah, that was really interesting, and I was wondering about the role of the pericytes in sprout behavior. So you show that the pericytes are positioned differently. So I was wondering, do you think that the pericyte is actively driving the sprout towards the capillary or the arteriole, or it's just following the decision of the sprout to reach towards the capillary or the arteriole?
I guess this is $1 million question, and this is really like in debating if it's the pericytes is leading or been leading. Looking to the ones that connect to arteries, they are really behind, and sometimes it's quite like a bit behind. So in that case, since it's leading, the endothelial cell is leading the kind of the parasites, but you also don't know how the connection and the mural cells, because they go to arterioles, they probably have some repulse because other studies in the lab showing that repulse in between parasites. So probably if they go there, they need to have something that repulse. But the ones that totally cover could be also driving, so could be the other way around. So I don't know if it could be some pathways or other marker there. We have some preliminary results that we observed EGFL7 that is present in the capillary ones and is not present at the ones that connect to arterioles. So could be also a question, could be also answered for that. So we have other signalings that perhaps could modulate the parasites or the other way around for that. But in this case, I don't think we can answer that so easily.
Great. I have another question, Vanessa. If you can comment on it, is a very general question, but how genetics or any environmental risk factor can impact the blood vessels development?
So we are doing now intermittent hypoxia model. So that is related to understand the apnea of prematurity. So it's intermittent. So we use from P2 until P12, and you observe the same way. And in fact, we observe this intermittent hypoxia can generate some sprouts from arteries. And I'm sure that, and it was the first time I was like really excited. I was like, oh, some sprouts from arteries and I'm sure if I didn't set up this groundbreaking work before, I probably will pass, oh, okay, they have some spots in arteries and some spots in the veins. But because now I know there is no way in the normal development that is spots from arteries. So I know that is something that could modulate. It could be because the hypoxia driving. Now we are trying to investigate how this environment change and this can alter the vessels and how is the wave, yeah.
Yeah, because I envision everything synchronized together during the brain development, and it's like a symphony. And if there is one factor can impact other developmental parts, the question like microglia, astrocytes, the lymphatic and the meningeal lymphatic site now, how all this really, the orchestra synchronized together during development, I think it would be super interesting.
Mm-hmm.
Within that, if there is no other question, we thank you so much again, Vanessa. It was a fascinating results and data, and we thank you again.